Hi everyone, I am Xuxu from Dr. Matthew Ellis' lab at Baylor College of Medicine. Thank you for coming to my presentation. Transcriptional reprogramming differentiates active from inactive ESR1 fusions in endocrine therapy-resistant metastatic breast cancer. Most breast cancers express estrogen receptor (ER) encoded by ESR1 gene. In breast cancers expressing Y-type ER, once bound by estrogen, ER internuclears form dimers and initiate transcription of estrogen response genes, thus leading to tumor growth. Endocrine therapies that inhibit ER function are highly efficient in treating breast tumors in this setting. However, many patients eventually acquire drug resistance after an initial response and develop metastasis to distant organs. One well-established mechanism is the point mutations in the ligand binding domain of ER. However, it does not explain all endocrine therapy resistance. Our group previously reported a novel mutational mechanism, ESR1 fusions, including ESR1 YAP1 and ESR1 PCDH11X fusions. They produce stable fusing proteins. In an estrogen-independent manner, they activate transcription of estrogen response and epithelium to mesenchymal transition genes, thus driving endocrine therapy resistance and metastasis. Now we know two fusions. Are there more ESR1 fusions? How clinically significant are they? We further identified 6 additional in-frame ESR1 fusions in metastatic breast cancer patients, shown by the circles plot. ESR1 gene forms fusions throughout the genome, including 2 categories: intrachromosomal, local translocation within chromosome 6, such as PCMT1 and ARID1B, and interchromosomal, fusing to distant genes on other chromosomes, such as YAP1, PCDH11X, and ARNT2. All 8 fusions follow the same structure pattern, containing the first 6 exons of ESR1, fused in-frame to C-terminal partner genes, thus excluding the ligand binding domain in ESR1. Therefore, the research gap is the diversity of partner genes creates a considerable diagnostic challenge in ESR1 translocated tumors. Due to the diversity of ESR1 fusion partners, I aimed to establish the rules that predict which ESR1 fusions are active drivers of drug resistance and metastasis. To functionally characterize these fusions, I expressed them in T47D, an ER-positive breast cancer cell, using lentiviral transduction. They produce stable fusing proteins, labeled by asterisks in Figure A. Since they lack the drug binding domain, an estrogen receptor degrader, fulvestrant, cannot degrade them. In comparison, endogenous Y-type ER are degraded. In addition to previously described ESR1 YAP1, PCDH11X, another 2 fusions with transcription factor partners, ESR1 SOX9 and ARNT2, drove estrogen-independent growth of T47D cells compared to the negative control YFP cells, shown by the blue bar. Other fusions, ESR1 DAP2, GYG1, PCMT1, and ARID1B, did not promote cell growth. I also included cells expressing truncated ESR1, the first 6 exons of ESR1 and cells overexpressing Y-type ER. They did not drive cell growth either. The growth driven by the 4 active fusions, as expected, was resistant to fulvestrant, shown by the yellow bar. To investigate the transcriptional profile driven by ESR1 fusions, I performed RNA sequencing on T47D cells expressing ESR1 fusions, as well as control cells in the presence or absence of estradiol, E2. Hierarchy clustering showed that T47D cells expressing ESR1 YAP1, PCDH11X, SONX9, and ARNT2 fusions clustered in the middle, distinctly from other ESR1 E6 fusions and control cells under E2-deprived conditions. All 4 active fusions demonstrated an expression pattern similar to inactive fusion or control cells treated with E2, consistent with potent hormone-independent transcriptional active of estrogen response genes, explaining the growth-driven phenotype of ESR1 fusions. Interestingly, active ESR1 fusion proteins also upregulated a cluster of genes that were not observed in the control cells, stimulated by E2, enriched with EMT gene sets. Consistently with activating EMT gene expression, active fusions, ESR1 YAP1, PCDH11X, SONX9, and ARNT2, promoted cell migration in an estrogen-independent manner, compared to YFP controls in scratch-owned assays in panel B. Cells were pre-treated with mitomycin C to inhibit cell proliferation. Cell monolayers were scratched and wound images were monitored for 72 hours. Cell migration were quantified by density in the wound area relative to that outside the wound area. Based on my study and publications, I summarized ESR1 fusions in ER-positive breast cancer into 3 categories: active fusions, cell context-dependent fusions, and inactive fusions. Active fusions, including YAP1, PCDH11X, SONX9, ARNT2, they can drive endocrine therapy resistance and metastasis. ESR1-DAP2 is a cell context-dependent fusion, driving hormone-independent growth in MCF7 but not T47D cells. There are also inactive fusions. Most of them were identified in primary tumors, although some fusions, such as GYG1, PCMT1, and ARID1B, from metastatic tumors produced stable fusion proteins, they are still inactive. From the summary, a functional rule is emerging. Interchromosomal translocations involving the first 6 exons of ESR1 gene fused inframe to 3' transcription factor or co-activator partners are active. However, ESR1 PCDH11X with a proton-cadherin partner is an exception, emphasizing the need to establish a gene signature to predict activity of ESR1 fusions. To develop a gene expression signature that is diagnostic for the presence of transcriptionally active ESR1 fusion, I used the RNA sequencing data on stable T47D cell lines, shown in previous slides, as a training set, to identify genes that were selectively upregulated by the 4 transcriptionally active ESR1 fusion proteins, as compared to 3 inactive ESR1 fusions and 3 controls. These two comparisons yield an overlapping group of 66 candidate genes with a fold change greater than 4 and a false discovery rate less than 0.5. These genes were enriched with estrogen response and EMT genes. I then devised an active ESR1 fusion signature based on estrogen response and EMT genes, as these were the top two pathways modulated by expression of an active ESR1 fusion protein. I further validated the signature in cells expressing additional ESR1 fusions, ER-positive PDX tumors, and the human cohort. In the training set, transcriptionally active ESR1 fusion proteins showed significantly higher scores as compared to inactive fusions and controls, as expected. To validate the 24-gene ESR1 fusion activity signature, I studied 7 additional ESR1 gene fusions, published by Priestley et al. In Nature. These in-frame ESR1-E6 fusions were identified in ER-positive metastatic breast cancer patients by whole genome sequencing. 3 fusions with transcription factor or co-activator partners, ESR1-ARNT2, LPP, and CoA1, were active, driving cell growth and motility. Surprisingly, ESR1-TCF12, which involves a transcription factor, was inactive. 3 gene fusions that involve protein-to-protein interaction domains, but without a transcription factor or co-activator partner, ESR1-CLINT1, GRIP1, and TNRC6B, were also active. These results emphasize again to establish a gene signature to predict activity of ESR1 fusions. I then performed RNA sequencing on T47D cells that expressed each new ESR1 fusion protein, as well as YFP control cells. 5 out of 6 active ESR1 fusions, ARNT2, LPP, NCOA1, CLINT1, and TNRC6B, demonstrated similar upregulated expression of the signature. ESR1 GRIP1 induced lower expression of the 24-gene signature than other active fusion examples. Interestingly, the 2 ESR1 point mutations, Y537S and D538G, induced similar levels of the signature as active ESR1 fusions, suggesting that despite different mutational mechanisms for ESR1 protein activation, ligand binding domain point mutants and translocated ER activate a similar pathogenic transcriptional pattern. The signature scores of active ESR1 fusions and point mutations were significantly increased compared to those of the inactive ESR1 TCF12 fusion and YFP control. As expected, the mean score of the weakly active ESR1 GRIP1 fusion fell below the cutoff value. The validation statistics of the independent Priestley et al. Set showed an accuracy of 90%. Since the 24-gene signature was similarly induced by ESR1 point mutants and active ESR1 fusion proteins, we named it MOTERA for mutant or translocated estrogen receptor alpha. To validate the MOTERA signature in human tumors, we annotated 20 biologically heterogeneous PDX models of ER-positive breast cancer. We grew them in 2 conditions, minus E2, shown by the blue curve, or plus E2, shown by the pink curve. Most PDX tumors expressing ESR1-YAP1 fusion or ESR1 point mutations showed A2 independent growth, with exceptions. Surprisingly, the growth of WIM40 was suppressed by E2. HCI013 expressing Y537S was totally E2 dependent. Most PDX tumors expressing Y-type ER showed estradiol-dependent growth. However, a few tumors demonstrated hormone-independent growth. We then performed RNA sequencing on these 20 ER-positive PDX tumors and detected the expression of MOTERA signature. As expected, on the most left in the heat map, the signature was highly expressed in WIM18 PDX, expressing the ESR1-YAP1 fusion. Endocrine therapy-resistant PDX, bearing a variety of ESR1 point mutations, also induced the MOTERA signature, confirming an overlap between the transcriptional properties of active ESR1 fusion proteins and point mutants. As shown in panel B, the MOTERA signature score was enriched in PDX tumors, expressing the ESR1-YAP1 fusion and ESR1 point mutations. Although the MOTERA transcriptional signature included estrogen response genes, expressing levels were not affected by E2 supplementation to those PDX. PDX tumors expressing ESR1 Y-type had significantly lower MOTERA scores in -E2 condition. The signature scores of Y-type ESR1 tumors increased with E2, consistent with some genes in the signature being estrogen response genes. Thus, as a screening tool, the MOTERA signature is more specific if the patient is taking an anti-estrogen. The MOTERA signature successfully distinguished between endocrine therapy-resistant tumors ESR1 fusion or mutations from ESR1 Y-type PDX with an accuracy of 95%. To further evaluate the performance of MOTERA signature in tumor samples, I used an independent RNA sequencing dataset of ER-positive metastatic breast cancer patients from the MET500 study. Signature scores were significantly elevated in tumors expressing ESR1 point mutations, such as Y537S and D538G, versus ESR1 Y-type samples. The two ESR1 fusions that I showed in previous slides, ARNT2 and ARID1B, were both originally identified from the MET500 study. As expected, the ESR1 ARNT2 fusion drove a high MOTERA score in the sample. The functionally inactive ESR1 ARID1B fusion also had a positive MOTERA signature score. However, this patient sample also harbored an ESR1 D538G mutation, explaining the positive score. In terms of performance, the MOTERA signature score significantly distinguished active ESR1 mutations from Y-type ESR1, with a sensitivity of 93% and a specificity of 78%. As a conclusion, this is the most comprehensive study to date to functionally characterize ESR1 fusions in ER-positive metastatic breast cancer. 10 out of 15 ESR1 fusions promoted A2 independent cell growth, motility, invasion, EMT, and resistance to fulvestrant. All followed a structure whereby the first 6 exons of ESR1 were fused in frame to C-terminal partner genes, replacing the ESR1 ligand domain. The diversity of partner genes creates a considerable diagnostic challenge, and no targeted treatments exist for ESR1 translocated tumors. The 24-gene diagnostic signature represents an efficient approach to screening samples for the presence of diverse somatic ESR1 mutations and fusions that drive endocrine treatment failure in metastatic breast tumors. Last but not least, I would like to thank Dr. Matthew Ellis and all our wonderful lab members, as well as collaborators and fundings. Thank you very much.